that I'm getting? Okay, so I wanted to talk about our muddiest points from last week, as well as sort of what's going on this week. Last week and this week are some of our more conceptual information, and that's a lot of what we will be going for forward. So this week we're doing cellular respiration, which is how our cells, or last week and this week, how our cells get energy. And then moving forward, we're going to be talking a lot about how cells use that energy for various processes. So before we get started on that, I found out that I can do something kind of cool, which I get very excited about. Let me do this really fast, which is kind of good as people are entering. But I want to share some files with you. So I want to share, I found out that I can share Okay, so you should see in the chat box that I shared the lecture. I also want to share this. Okay, this takes a little while. So if you look at the other file I shared, I'll also pull it up on my screen. I wanted to mention that we just got official approval. We've been working very hard to do this, but we just got official approval for having a presence of the Pitchfork Pantry on the West Campus, which is very exciting. So Pitchfork Pantry is our food pantry associated with ASU. And so this is the flyer I shared with you. I will also send it out in a message. But I do want to mention that if you would like to pick up some non-perishable food items, this is just a sample of what we offer at the pantry. And you can sign up, so it'll say scan me. You'll just sign in with your ASU email address, and you'll be qualified to do this, to grab a box. And you'll notice that there is a pickup on the West Campus. Again, this is brand new. I'm like very excited. It took a lot of fighting to get this on West Campus and not just at Tempe, but we're going to have presence of this pantry on all 4 campuses. So again, I will share this with you again at the end of class. I will send a message out as well. But on West Campus, the pickup location is in the Fletcher Library, lower level in the Writing Center, so sort of around the Tutoring Center. So I'm very excited to announce that, and we'll be having more. So this is just for signing up for the October pickup. And I do know that items have recently been replenished. We had a drop off of supplies today. So I just want to share that because, again, if you're an ASU student, you know, you're qualified to receive this. So no questions asked. We wanted to make sure that there were some resources. Another very exciting thing we were just awarded, actually our student representative, who is Narjish, was just awarded a grant to, in conjunction with the food pantry, we're gonna have like a closet for professional clothes for like interviewing and stuff like that. So that is forthcoming, but the pantry exists right now. If you are interested in being a part of the pantry or volunteering, reach out to me and I can put you in contact. I am the faculty advisor for the Pitchfork Pantry. So if you have if you want to be involved, we would love to have you involved, and I can put you in contact with the people who would facilitate that. We'll be having pop-up events on campus as well. So that is that. Again, I'm very excited to share those news. Been waiting a long time to get that on the West Campus. So makes my heart happy to know it's finally here. Okay, so now let's get into Muddiest Points. I want to honor your time and talk about this and make sure that you get the information you need, which is very important. I do want to mention that if you have questions or comments or anything like that, I have the chat open, but also feel free to unmute yourself and speak out. Anything you want to do is fine with me. I am willing to bet we will not get through my entire lecture today. So I will be available having class open on Friday as well. So yeah, pop in and we can finish this, or if you feel comfortable, you know, that's your prerogative, but I will record it regardless. Okay, so let's get started. All right, so just to give you an idea of, I know that it's Wednesday, so you probably have a pretty good idea of what to expect this week, but just briefly give you an intro to what to expect this week in Bio 181, lab and lecture, and then we'll go through some Muddiest points. This week is a relatively light week. You do have lectures over anaerobic cellular respiration and photosynthesis, so hopefully you're going through those. My hope is that they will also be supportive of the information you learned last week, which was cellular respiration, kind of to reiterate those concepts and sort of apply them in a different way. This is Unit 3, so we wanted to focus all your attention on energy generation for an entire unit. So I will be very soon posting another review and checkpoint. So I'll be posting that Friday, and you will have, again, a week to study and review before the checkpoint opens. So I'll send out all that information soon, because this week actually culminates our next unit already. So yeah, all right. In lab, again, several of you have probably already had lab. You do have some individual assignments. There are 3 labsters this week. One is on experimental design. One is the electron transport chain, which is very kind of going over an electron transport chain pertaining specifically to photosynthesis. So supportive of those concepts. I know the labsters are really weird, a little alarming simulations, but I noticed somebody commented in the discussion board that there was a lot of, you know, obviously confusion with cellular respiration, but the comment was that the labster that covered cellular respiration did a really good job kind of going step by step through the process. So I know that they're a little alarming, just like the way the people talk and look in those simulations, but I think that the concepts are so helpful. So, you know, take your time through those, don't rush. They are very good, useful tools. One of the labsters this week is on micropipetting, which is actually going to be the focus of our next round of on-campus lab opportunities if you want to come. So hopefully you've heard from your lab instructor this week, asking for you to contact them by the end of the day today. So by 11:59 p.m. this evening, please just reply to your lab instructor saying, yes, you will be there, or no, you won't. Those labs will focus on micropipetting, which is a very important technique for biology, not only kind of like technique-wise how to micropipette, but also pipetting accuracy. So even if you have experience with micropipetting, a really important thing is pipetting accuracy. So very helpful in that way. Those are optional opportunities, ungraded, so do not feel obligated to come if you cannot. If you want to come, we would love to see you. If you don't feel comfortable coming to campus, totally fine too. As I've mentioned previously, my hope, like, I'm gonna make sure that these opportunities are present in the future once things settle down, to make sure that you can gain these opportunities as well. So no worries there. Aside from that, this week you do have some lab work pertaining to the enzymes experiment, which you started next week. So that continues for the next couple of weeks. This week you're working on your methods and results, collecting feedback from your instructor, and then their final drafts of those are going to be due on Sunday. And then just meet at your scheduled lab time. All right. And again, please, if you are wanting to come to campus for those on-campus labs, please let your lab instructor know by the end of the day today. Okay. Did I hear somebody?
Just a quick question.
Oh, yeah.
I just said, quick question about like.
Masara, was that you?
Sorry, I got cut off. Sorry.
Oh, okay. I was like, I heard you and then it stopped. Okay.
Yeah. So when we're collecting the data, are we gonna have like an average this time or not? Because I mean, since we're measuring like, the effects over, specific independent variables, it wouldn't make sense to have an average. Am I correct?
That would be a question for your lab instructor.
But okay.
Yeah.
Okay.
Sorry.
Okay.
I don't, as you probably have noticed, I'm only teaching lectures this semester, so I'm a little out of the loop when it comes to like the exact lab stuff. It just has to be the same.
No problem. It's okay. I thought maybe, you would,
So I wish I knew what was going on. I miss teaching lab. But although I love teaching you all in lectures, so that makes me happy. Okay. So, yeah, and I would say that going forward too, if you have questions about lab, make sure to reach out to your lab instructor just because they'll know so much more than I do. I'll try to help as much as I can, but I'm kind of out of the loop when it comes to lab stuff. All right, so let's talk about what I am in the loop with, which is lecture content, which I love. So I know, like when I read through the Muddiest points, a lot of people were like, wow, this is a lot. I couldn't agree more. But also I do want to mention that cellular respiration is one of, I think, the most difficult concepts in Bio 181. I think that your progression as a scientist will be to really not enjoy it right now. And then you'll really not like it the next time you see it if you pursue biology. And then by the fourth time you see it, because yes, you will see it in every single biology class you ever take, by the fourth time you see it, you'll be bored with it and you'll be like, okay, which actually makes you enjoy it because you're like, I've seen this before, I know it. I love cellular respiration. I think it is fascinating. I think it's amazing. So that's I guess it took me a long time to get there though. I would say that it took me teaching it to actually enjoy it. So it's okay if you don't love it. That's okay. I get it. I was with you too. I do want to mention that like kind of first thing, and I'll zoom ahead right here. When I looked through the discussion board, a lot of the comments were sort of like, I don't understand the Krebs cycle, or I don't understand pyruvate oxidation, something like that. And when reading through those comments, sort of the thing that I was noticing is that I think a lot of the confusion came from two things. One is that the textbook goes into excruciating detail about these processes, and you don't need to know that much detail for our class. So this is a really good example of why the textbook reading is always, always, always going to be supplemental. It'll give you a good idea of some like test questions or homework questions that you could anticipate seeing. But it's always just going to be supplemental and supportive of the content. The material you should focus on, like the material you need to know, is the material that's presented to you in the lectures. So just a reminder, when it came to like the Krebs cycle in the lecture, I had this little box that was like, you don't need to know all of these steps. So always pay attention, really, really, really pay attention, especially moving forward in this class. Focus on what's in the recorded lectures. That's what you need to know, and that's the level at which you need to know it. That said, we still covered a lot in those recorded lectures. So I'm going to go through today, I do want to go through substrate level versus oxidative phosphorylation, which will give us a good review of redox reactions, which will also lead us into cellular respiration. My guess, though, is that cellular respiration, like what do you need to know, will be probably have to take place on Friday. Again, if you can't make it on Fridays, I'll record it as well. So just like today and every day. Okay, so let's go through it. So our Muddiest points again, I did see a lot of comments saying I'm confused about ATP generation, redox reactions, especially I felt for some of you that were like, I thought I understood these things and then the textbook explained it and I was confused. I feel you. And then cellular respiration. The main question was cellular respiration, like the Krebs cycle. What do you need to know? And what my answer to that is, you need to know a lot, but not that much. All right, so let's talk about exactly what you need to know. Okay, so first off, I do want to mention, we do have we did discuss in those lectures, 3 ways of making ATP. And again, I want to reiterate, anytime I'm going through this stuff, if you feel lost or confused, let me know in the chat box, and also feel free to unmute yourself and reach out. Okay, I know that this is a lot of technical stuff, so if I need to slow down or back up, I will. Okay, I want to make sure that you get this. Understanding this conceptually, if you understand cellular respiration conceptually, you will be like so happy for the next steps in biology. It will make your life so much easier, trust. Okay, so there are 3 methods of making ATP. And the primary difference is, or at least in my mind, how I like to sort of like divide them, is asking myself, where is the energy coming from, or the phosphate coming from, to create the ATP? So last week we talked in depth about the first 2 types of ATP, or methods of making ATP. We talked about substrate level phosphorylation, and we talked about oxidative phosphorylation. So in this week, when we're covering anaerobic cellular respiration, fermentation, and photosynthesis, you'll reiterate these concepts. But photophosphorylation is pretty much specifically photosynthesis. So photophosphorylation is what happens in photosystem 1 when energy from the sun splits water, and those electrons lead to an electron transport chain, which generates a proton motive force, which generates ATP. So that's photophosphorylation, is essentially the events of photosystem 1. So that's covered in this week. But since today we're talking about last week's material, cellular respiration, I want to review substrate level phosphorylation and oxidative phosphorylation. So the 2 that specifically pertain to cellular respiration. So let's start with substrate level phosphorylation. So first off, just to kind of emphasize, like, you can sort of understand like where the energy is coming from for this process if you understand what it means by substrate level. So in order to understand that, just like showing this picture again, this picture is showing an enzyme. And remember, enzymes facilitate chemical reactions. And remember, enzymes facilitate chemical reactions. You have reactants or substrates that enter reactions and products that exit. So substrate and reactant are essentially the same thing when it comes to enzymes. So here, your reactant or your substrate is what would fit in the active site of this enzyme. And in substrate level phosphorylation, this enzyme is going to basically steal a phosphate from a substrate and then pop it onto ADP. So it's called substrate level phosphorylation because right here at the level of the substrate, the substrate is actually donating the phosphate. So really importantly, if you think about the steps of cellular respiration, we talked about glycolysis and acetyl-CoA or pyruvate oxidation and Krebs cycle. And in each of those steps, we were generating, releasing electrons, generating NADH, and using that NADH to donate electrons at the electron transport chain and then generate ATP. So multi-step process for ATP generation. That is not substrate level phosphorylation, or at least anytime you're generating NADH, that means it's not substrate level phosphorylation. Substrate level phosphorylation is when you literally take a phosphate off of a substrate and pop it onto an ADP. So this does happen throughout the process of cellular respiration. One very kind of like clear place. So I'm going to pull up the slide for glycolysis. And again, I realize this is a lot. I'm going to pull it up. Substrate level phosphorylation does occur during glycolysis. So this is the process of glycolysis, those 3 steps. And I want to draw your attention to the fact that we start glycolysis with a 6-carbon molecule, glucose. And you'll notice we use 2 ATP. We actually pop the phosphate off of these ATP and pop it on this 6-carbon molecule. We phosphorylated this molecule. This is substrate level phosphorylation. It's just not generating ATP. But this process where we took a phosphate from here and added it to this molecule, that's substrate level phosphorylation. It's not only used to generate ATP, but it can be. So in the first step of glycolysis, we do substrate level phosphorylation, not to generate ATP, but actually to generate the 6-carbon phosphorylated compound, which very quickly splits into glyceraldehyde 3-phosphate, or 2 molecules of glyceraldehyde 3-phosphate. So you see glyceraldehyde 3-phosphate, or G3P, is a 3-carbon molecule with a phosphate. We go through another round of substrate level phosphorylation, and now we have 2 3-carbon molecules, each with 2 phosphates. And you'll notice actually during what's called the payoff stage of glycolysis, we actually, ignore this part, we actually steal these phosphates. So in the process of the payoff stage, we're taking this molecule and we're going to steal these phosphates from this molecule and add them to ADP in order to make ATP. So again, anytime we're adding a phosphate to something, that is substrate level phosphorylation, right? It's happening right here, it happens right here. Substrate level phosphorylation can phosphorylate anything, but it is one method that can be used to phosphorylate ADP into ATP. And again, that's what happens here. So you have this 3-carbon molecule with 2 phosphates. We pop those 2 phosphates off, and they're used to phosphorylate ADP into ATP. So during glycolysis, this is one stage of cellular respiration where we get some ATP generated via substrate level phosphorylation. We used these phosphates, added them to ADP, and got ATP. Here is the thing that I think is often confusing. Okay, so we do get some ATP via substrate level phosphorylation during glycolysis, but we also generate NADH. This is unrelated reactions. So anytime you see an arrow like that, that's indicating a reaction took place. This arrow right here is indicating that substrate level phosphorylation happened, phosphorylating ADP into ATP. This arrow right here is indicating that a bond was broken and electrons were released, and those electrons were bound up to NADH, okay? So I want you to just shout it out, all right? So unmute yourself. Is NADH energy that we can use for reactions? So just yell yes or no.
Only if it's converted to NADH.
What? I heard something, but I didn't have my volume up loud enough.
Only
Can you say
when yes, when it's converted to NADH, then we can use it for the proton motive force.
So to a stat, exactly. So everybody, hopefully you heard Masara. So is NADH energy? NADH itself is not energy, but Masara said only if it goes to an electron transport chain to generate a proton motive force. Okay, so this is very important because if we look at this, right, in glycolysis, we do get some ATP, which can be used right away for energy. ATP is energy immediately. If we get NADH, that is not immediate energy. This NADH, right, that's created through oxidation, right? We break a bond, release an electron. That means we oxidized to this molecule, reduced NAD into NADH. But then NADH has to travel to an electron transport chain to be converted into ATP. It's not energy itself. It has to travel somewhere to be turned into energy. So I mention that because if you see NADH produced, that means that will lead to ATP produced via oxidative phosphorylation. So we'll go through that in a minute again, just to review what oxidative phosphorylation is. But if you see NADH generated, which you do right here during glycolysis, that tells you that ATP will eventually be generated via oxidative phosphorylation. We also see another reaction right here where we get ATP generated right there. That is ATP generated via substrate level phosphorylation. Okay, so I know that can be a little confusing, right? We're looking at glycolysis, but there's two ways that ATP is generated during glycolysis. The first way is substrate level phosphorylation, where we get some ATP right away. The second way that energy is generated during glycolysis is by oxidizing that organic compound, releasing electrons, reducing NAD into NADH. But importantly, so that's oxidation, but then that NADH has to travel to an electron transport chain in order to create ATP. So remember, substrate level and oxidative phosphorylation are just ways that we can generate ATP. There are reactions with multiple steps, like glycolysis, where we will get ATP generated in multiple ways. So again, glycolysis, we do get some energy generated, some ATP generated right away by substrate level phosphorylation, but we also release electrons, which can eventually lead to ATP production via oxidative phosphorylation if there is a corresponding electron transport chain. So very important. Okay. Can anybody tell me another place during cellular respiration where we have both oxidative phosphorylation and substrate level phosphorylation? Unmute yourself, you can raise your hand, do whatever you want. Where's another reaction through cellular respiration where you see ATP generated and NADH and FADH2 generated?
Is it the Krebs cycle?
Yes, good job, the Krebs cycle. Let's look at the Krebs cycle. This is the Krebs cycle, okay? Notice we get some NADH that has to go to the electron transport chain, so that would be oxidative phosphorylation. But look down here, we also get ATP. This ATP is generated via substrate level phosphorylation. So glycolysis and the Krebs cycle are both able to generate ATP via substrate level phosphorylation and oxidative phosphorylation. So my general rule, if you're like, well, how can I determine like how ATP is generated? My answer is you gotta look at the reactions. But if you just see ATP just kind of like randomly appear, like if it's just like, here's a reaction, ATP, it's probably substrate level phosphorylation. If you see NAD being reduced to NADH, or another electron carrier like FAD being reduced to FADH2, and then a subsequent electron transport chain, that's oxidative phosphorylation. Okay, so if you see ATP, just substrate level. If you see NADH or FADH2 or NADPH, that is oxidative phosphorylation. And again, you can see the Krebs cycle, it's a lot, but at the minimum, you can say, okay, that's NADH, that means ATP via OxPhox. NADH, that's gotta be oxidative phosphorylation. NADH, FADH2, that's gotta be oxidative phosphorylation. Oh, ATP, substrate level phosphorylation. Same here, again with glycolysis. I see some NADH, it's gotta be oxidative phosphorylation. I see this reaction, ATP, it's gotta be substrate level phosphorylation, okay? So look for, if there's multiple steps, if it requires an electron transport chain, it's oxidative phosphorylation. If it doesn't, it's substrate level phosphorylation. Okay. So that is the main difference between substrate level and oxidative phosphorylation, just like identifying or kind of contrasting the two. Again, substrate level phosphorylation is just when we take a substrate, pop a phosphate off and use that to phosphorylate ADP into ATP. Oxidative phosphorylation is a multi-step process. So first off, oxidation is when you have one molecule that has a bond in it or carries an electron, right? And that electron is lost. So you see A lost its electron. That is oxidation, when a molecule loses an electron. Reduction would be a molecule gaining that electron. So something to note about this is that electrons by themselves, so if you had a molecule and it released an electron, electrons by themselves can be very, very damaging to a cell or to a system. So if you've ever heard of like oxidative stress or oxidative damage, if you think about it, like bonds are broken and bonds are created by electrons. So the negative charge kind of just like floating around can break bonds, which would just like destroy structures in a cell. So this is why we don't ever just have like an oxidation reaction take place. If a molecule is oxidized, meaning it loses an electron, it will have a paired molecule that binds up or gains that electron. Because again, cells and systems don't ever want just like random electrons floating around. If a molecule loses an electron, you're going to have another molecule gain that electron, because we don't want to let them just sort of like hang out. So redox reactions are those paired reactions where you have oxidation, loss of an electron, and a corresponding reduction or gaining of an electron. One of the kind of confusing terminologies associated with this is reduction, right? So like, why, if this molecule gains an electron, is it said to be reduced? And the answer is because electrons are negatively charged. So what's happening to this molecule by gaining an electron, its charge is reduced. So that's why we say molecules that are reduced gain an electron, because they're gaining a negative charge. So their overall charge is being reduced. So those are redox reactions. I was taught to like remember like what oxidation or reduction is. I was taught oil rig, like oil oxidation is loss, and then rig reduction is gain. But a colleague of mine prefers Leo the lion says grr, which is fun. But I did this terribly because I did it randomly yesterday. But Leo the lion says grr is another way to remember it, where Leo is losing electrons is oxidation, and then gaining electrons is reduction. I don't care how you remember it, but you do need to remember it. You're probably co-learning this in chemistry if you haven't already. So that's the basis of a redox reaction. But again, we talked about substrate level phosphorylation. And now what I want to move into is oxidative phosphorylation. So we have a couple minutes. Left. And so I want to spend that on oxidative phosphorylation. And then if we have any time, I'll just kind of like briefly go through kind of like what to focus on cellular respiration. Although again, we probably will need to meet on Friday if you can, right? If you can't, I understand. So oxidative phosphorylation is when we have these redox reactions where you have a molecule that is oxidized, meaning electrons are released, and then you have a corresponding molecule that is reduced. And so cellular respiration is such a good example of this. In cellular respiration, what's happening is you're oxidizing an organic compound, releasing an electrons from that organic compound, and those electrons are bound up by NAD or FAD, not NADPH in cellular respiration. That's used in photosynthesis, but same idea. These molecules are reduced, and so they then carry those electrons to the second part of oxidative phosphorylation, which is when those electrons are then used to generate a proton motive force. So first off, just to kind of look at redox reactions. So at least in a metabolic pathway, remember that chemical bonds are formed via interactions really with electrons, right? So chemical bonds are formed between, at least like we're primarily talking about covalent bonds, which would involve the sharing of electrons to basically fulfill the orbitals of either of these molecules. So essentially we kind of think of chemical bonds as like methods of like storing electrons. And so what happens is if we break this bond, right? So here's my enzyme breaking this bond. What happens is an electron is released. Okay, so again, that's oxidation. Because we have a bond in this molecule, that bond is broken, an electron is released. Again, we don't want electrons just floating around in space because they're negative charges and they can tear stuff up. And so what we do is if we release an electron, so if this molecule is oxidized and we release an electron, we bind that electron up by reducing a corresponding molecule. There's always going to be like NAD or FAD. And when NAD gains that electron, it gains a negative charge. And so now it is not neutral. And so what these reduced molecules do typically to kind of like neutralize their charge is if they gain an electron, they typically also gain a hydrogen, which is why when NAD plus or FAD plus gains an electron, you can tell if it looks like NADH, because again, that H is talking about the fact that you gained a hydrogen ion at the same time as gaining this electron. Those hydrogen ions are also very important, as we will talk about at the electron transport chain. So again, redox reactions, such as those that occur in cellular respiration, we're breaking a bond in a molecule, releasing electrons from those bonds. So that's oxidizing this molecule. A corresponding molecule is reduced. It also gains a hydrogen to neutralize its charge. So what do we do with those electrons and those hydrogen ions? Well, again, NADH itself is not energy. We want to use NADH to generate ATP. And we can do that at an electron transport chain. So what happens here, I'm actually going to switch ahead to the next slide. What happens here, here's exactly what we looked at just now. We have this molecule, a bond is broken, electron is released, so you have this molecule oxidized. the corresponding molecule is reduced. So you see it gaining an electron and a hydrogen to become NADH. That NADH then travels to an electron transport chain, and then actually it's oxidized, right? So NADH is oxidized, meaning it releases its electrons. It also releases corresponding hydrogens. And those electrons are used as energy to pump hydrogens to one side of a membrane. So if this is in eukaryotic cells, this would be to one side of the mitochondrial crista. If this is in prokaryotic cells, this would be to like one side of the plasma membrane, and they'd be trapped by like the cell wall. So again, NADH travels to the electron transport chain, uses electrons to pump hydrogens to one side of the membrane. So one of the points of confusion that I noticed in the Muddiest point discussion board was the term proton motive force. Like, why is this called a proton motive force? And why is this energy? So in order to understand the term proton motive force, what you need to understand is a little bit about hydrogen. Hydrogen, it actually doesn't have any neutrons. So it essentially in its nucleus has a proton, which remember are positively charged. And then, you know, to balance that out, it has one electron. So technically hydrogen atoms, because they don't have neutrons, have one proton and one corresponding electron. So notice this is a hydrogen ion, which means that it actually lost its one electron. But if you have a hydrogen ion that loses one electron, that means that the only subatomic particle it has left is one proton. So sometimes you'll hear hydrogen ions referred to as protons, because that's like all they are, right? And so what happens, again, is you use the energy from these electrons. These electrons are used as energy to pump hydrogens to one side of a membrane. This is called a proton motive force, because again, these are technically just like a ton of protons on one side of a membrane. So why is this energy? And this is when you got to think about the fact that concentration gradients are a tremendous amount of potential energy. So I tried to frame this sort of like where it would look similarly. So if you think about this membrane as sort of like this dam, and these hydrogen ions like the water behind the dam, all of this water stored behind this dam like wants to flow, right? It's being held back. But if you can imagine what would happen if this dam broke, all of that water would fly past, right? It'd be a ton of energy, right? That's my best impression of water flowing. But if you broke this dam, all this water would come gushing through. And that's the same concept here, right? It's exact same concept here. We're pushing all these protons to one side of the membrane. They're held back behind that membrane. But what that means is if they're held back there, that's a ton of energy. And you'll notice that in the membrane, we have ATP synthase, which is an enzyme that creates ATP. And that proton motive force, all that potential energy behind that membrane, is what is used to fuel ATP synthase. So ATP synthase is kind of interesting. It's actually kind of very similar to like hydroelectric power or like a revolving door. Or if you can picture, or like one of those, you know, those wheels where water flows into it and the water and the wheel turns. That's very similar actually to how ATP synthase functions. You can't see it here, but it basically has a wheel where every time one hydrogen kind of pushes through this enzyme, you get enough energy for ATP. So that's why we say one hydrogen will lead to one ATP. But again, this enzyme has to have energy to make ATP because that's a high energy bond, phosphorylating ADP to ATP. You're creating a high energy bond. And that's where you get the energy for that bond. Again, you can create ATP via substrate level phosphorylation. That is very true. But I want to remind you, if you think about cellular respiration, in total, you get 4 ATP from substrate level phosphorylation. You get a net total of 2 during glycolysis. You do generate 4, but because we have to use 2 in glycolysis, we say we get a net of 2. So we get 2 from glycolysis, and we get 2 total from the Krebs cycle. So that's 4 ATP total from substrate level phosphorylation during cellular respiration. But via oxidative phosphorylation, where we break bonds in glucose, we break a bunch of bonds in glucose, we release electrons from all of those bonds, we use those electrons to reduce NAD or FAD into NADH or FADH2, which will then travel to the electron transport chain. Each of those, like NADH, can pump 3 hydrogens to one side of the membrane. FADH2 can pump 2 hydrogens. That means that, yes, we get 4 ATP via substrate level phosphorylation, but it ends up being 34 from oxidative phosphorylation. So that's where we get our total of 38. 4 of those ATP are generated via substrate level, but 34 are generated via oxidative phosphorylation. So yes, substrate level phosphorylation is like way easier, but oxidative phosphorylation is you get so much more ATP via oxidative phosphorylation, which is kind of the rationale for going through the whole process. Okay, so we have just a couple more minutes. I noticed a couple comments in the chat. I also noticed that Noor, you have your hand raised. So Noor, do you want to go ahead?
So I was just gonna ask, so there's like all these types that you're talking about, they're like steps in order. So altogether at the end, you release 34?
So I actually have a slide about this. So when it comes to the different types of ATP generated through this process, so I wanted to do this together. So the PowerPoint that I shared with you, which if any of you came in late, I did post it on earlier, but I'm gonna post it again because I want to make sure that you can do this. Just takes me a minute. Okay, so you should see it in the chat. So what I wanted to do together is exactly that, right? So you all saw this table telling us in glycolysis, we got 4 ATP, net of 2, via, right there at the reaction. We also got 2 NADH, which after the electron transport chain would give us 6 ATP, etc. But what I wanted to do is ask, right? So like, okay, well then via what method? So remember, via glycolysis, if we go back, we do get 2 NADH, okay, but we also get those 4 net 2 ATP specifically via substrate level phosphorylation. So these 4 ATP net 2 were generated via substrate level phosphorylation, whereas those 2 NADH will eventually contribute to the production of 6 ATP via oxidative phosphorylation.
So just to make sure I'm understanding, this is like cellular respiration, and then there's like the cellular respiration and fermentation, and then these are the steps for cellular respiration, but then fermentation is just the glycolysis step.
Oh, exactly. Yes. So this, right, we've only talked about today, well, I guess what I should say is today we were talking about all the steps of cellular respiration. But fermentation, you are correct, is only glycolysis. So that's why glycolysis, its output of energy, because you don't have an electron transport chain, the only energy generated via glycolysis is via substrate level phosphorylation. So that would mean you would only get 4 or net 2 from fermentation.
Okay,
Does that
thank
make
you.
sense? Yeah, good question. Yeah, so exactly, like that's a really important point. So the chart we're looking at right here is for cellular respiration. So I do want to do this chart, but I do realize like we are out of time. So if you do have a second to fill out this chart, again, I am recording it, but if you do need to go to another class, that's totally fine. I'm going to continue filling out this chart for a minute, and then I will also answer the questions in the chat. But again, it's recorded, and if you need to go to another class, I totally understand. I want to respect your time. So let's go through this chart. And again, I will post this on Canvas. So we got ATP, substrate level phosphorylation. We got NADH, which will eventually lead to ATP. So that tells us oxidative. More during pyruvate oxidation, we got 2 NADH, which will, at the electron transport chain, give us 6 ATP. Because it's NADH, it's oxidative. Looking at the Krebs cycle, we did get some ATP just like generated right there. Because it didn't lead to NADH and then ATP, this tells us it's SLP, or substrate level phosphorylation. And then we got NADH and FADH2. Both of those are used for oxidative phosphorylation. So this tells us that we got 4, but net 2 via substrate level phosphorylation during glycolysis, and 2 via substrate level phosphorylation in the Krebs cycle. So that would tell us we have 4 ATP generated via substrate level phosphorylation, and then the remaining 34 bringing us to our total of 38 via oxidative phosphorylation. I think that was one of those questions on the quiz that some of you were asking about. So I wanted to be sure to address that. So some questions in the chat. How do you tell the difference without a diagram for the, so for example, if I was saying like there is 10 ATP generated during this reaction.
It's for the Krebs cycle and also the other diagram that was shown when you said, okay, if you see random ATP generated, you know that it is this thing. If you see NADH, but like in like tests and stuff like that, you don't get those diagrams. So how are you able to like tell the difference?
Yeah, so like on a test, for example, I would like a question that you could anticipate would be something like how much ATP is generated during glycolysis via substrate level and via oxidative. So you would have to know, like use your knowledge of that pathway, but I would never like say, here's a reaction, like how is ATP generated, without you, without you having the explicit learning objective of learning that reaction. Does that make sense?
Yeah.
Okay, so yeah. And I guess like we didn't discuss necessarily, yeah. And I guess short answer to your question is like, you couldn't, right? So you would have to see the reaction to determine how ATP is generated.
Okay, you just like, okay, because you told us that we have to identify through looking at the diagram, and I was just thinking like, if I didn't have that diagram, like how was I supposed to, identify it? But yeah, you answered my question.
Okay, good.
Thank you.
That's a good question. Yeah, thanks. See, and then, yes, I can definitely go back to the terms for remembering them. I also posted this lecture in the chat, and it is on Canvas. So you should have all of these slides as well. So all of this is available to you. Humanistic, what is used instead of oxygen in anaerobic cellular respiration? And is it just as efficient? And the answer is, in anaerobic cellular respiration, honestly, there are a variety of final electron acceptors. So it just depends. So in the lecture this week, I talked about a couple just as examples, like sulfate or nitrite, sulfite and nitrite, or sulfate and nitrate, I can't remember. But those are some examples of alternatives to oxygen for a final electron acceptor. You can use like iron, you can, honestly, I probably couldn't list all of the alternatives because there are so many organisms out there that use such a wide variety. So yeah, and so one of the reasons, like, is it just as efficient? Short answer is it can be pretty close. So if you remember, like with aerobic cellular respiration, you could generate like up to 36 ATP, whereas aerobic cellular respiration, you can generate 38. The big difference is that, and we didn't go into depth about this because you don't need to. But for example, like if you were to do cellular respiration and not have oxygen, you would need to make sure that the cell had the ability to use a different final electron acceptor. And sometimes just using a different final electron acceptor requires some extra processing, which would require some ATP. So it's not that the steps generate less ATP necessarily, because they're the same steps. It's just that you would have another step that like required ATP, sort of just making your overall output less, if that makes sense. It's an excellent question. All right. If there are any other questions, I'll be happy to hear them. If not, you're good to go. And if you have time, I'd love to see you on Friday, but if you don't, I understand. I will record the lecture no matter what. All right.
Thank you.
Thanks so much. Have a wonderful day. Bye, everybody.